Welcome to today's Adelaide BioMed Mini Reviews webinar series. My name is Yvette van Eenennaam and I'm the General Manager of Adelaide BioMed, the Health and Medical Innovation District in the heart of Adelaide, what you see in the background. And I'm also pleased to be today's chair. Before I introduce our first speaker, I would like to acknowledge the Kaurna people, the original custodians of the land we meet on today. And I would like to pay my respect to the elders, both past, present, and future. So these weekly short webinars are organized to encourage further collaboration within our innovation district, but also with local, national, and global parties. So collectively, we're really able to transform Adelaide into a vibrant, innovative health and medical ecosystem, and great things are currently happening in there. So this afternoon, we're very fortunate to have two great speakers, Dr. Kurt Seals from Agilex Biolabs and Mark Roe, CEO of Fusetec. And our first speaker today is Dr. Kurt Seals. And Kurt is Director of Immunoassay and Immunobiology at Agilex Biolabs. And I'm not sure if everybody's aware, but Agilex Biolabs is the largest regulated bioanalytical laboratory in Australia, with its headquarters here in Adelaide. And they recently had a new 1.5 million facility, a vaccine and immunobiology facility. And I believe it's the most sophisticated one in the Asia Pacific area. Attracts biotech pharma from around the world for advanced clinical research and has over 20 years of experience in early phase clinical trials with an excellent track record of enabling movement of new drugs entities to market. So thank you, Kurt, for joining us today, and it's a pleasure to hand over to you now.
Thanks very much, Yvette, and thanks very much to BioMed City for the invitation. And hello, viewers, and hopefully today I'll be deconvoluting a little bit about the bioanalysis for clinical trials. So the clinical ecosystem is quite complex. It comprises multiple different stakeholders, consisting of laboratories, CROs, hospitals, phase 1 units, and other service providers. And cumulatively, it provides an ecosystem for the successful clinical trial scenario. And Agilex Biolabs fits into this ecosystem as part of regulated bioanalysis, and we provide the pharmacokinetic and pharmacodynamic measurements for the novel drugs that are coming through to the market that are then being dosed into these either healthy volunteers or patient populations. So the clinical trial ecosystem really encompasses a fast startup in Australia. So everything sits within the ethics committee in Australia, and they review all of the preclinical pharmacokinetic data, the safety data from the animals, any in vitro testing that had been done on that novel drug or novel chemical entity that is coming through to market. And they evaluate this entire package as part of an investigator brochure and determine whether it's safe to move into those either healthy volunteers or patient populations. So the clinical trial itself is really managed by the CROs together with all of those other service providers. And we're an add-on specialty laboratory that provides those bespoke pharmacokinetic and pharmacodynamic tests. So what are we? We're Agilex Biolabs, really the largest bioanalytical laboratory within Australia. Interestingly enough, we're a success story for South Australia. We spun out of the University of South Australia way back in 1996, as you can see on the left-hand side history column. And over the years, we attained accreditation. So we're NATO, OECD, GLP, and 17025 accredited. And we really are structured to the three main service lines, which is toxicology. So we can do small rodent GLP toxicology, and then partnering with other labs overseas to provide second species. And then small molecule bioanalysis by tandem mass spectrometry, and then large molecule analysis by ligand binding assays and other classical assays to support immunoassay and immunobiology analysis. And then we can look at functional and PD biomarker endpoints as well. So what do? We're providing an end-to-end solution for bioanalysis. And this slide is very, very busy. And I urge you to just not worry about taking notes and have a look at it offline at your own leisure. But really, we provide a full service regulated bioanalytical service from preclinical toxicology, working with early discovery companies and university spin-out groups and university labs that are looking at putting their non-GLP and GLP tox into a regulated environment in order to provide proof of concept data to progress their molecule up to the clinic. PK assays, immunogenicity assays, neutralizing antibody assays, and then pharmacodynamic assays, which are either immunological assays by flow cytometry or other bespoke assays that are really geared towards understanding what the drug is doing. So PK and PD are basically what the drug does to the body. So for PK, that's the absorption, distribution, metabolism, and excretion part of the drug cycle. And then the PD component is whatever the body is doing to that drug. So either developing an antibody against that drug or switching on a pathway within those cells and causing a cytokine or chemokine to be made. So we measure new chemical entities, so new drugs that are being developed either for brand new indications or orphan indications, new biological entities. These might be proteins, monoclonal antibodies, bispecifics for, say, immuno-oncology. We also deal with vaccines and other gene therapy products. We measure anti-drug antibodies as a consequence of developing those large molecule therapeutics, and then a whole variety of immunological assessments as shown on this slide. So how do we measure it? Well, we use a variety of sophisticated equipment. Many of the equipment on this page you'd be familiar with being in the academic setting. They're quite common platforms. In a regulated environment, we would use the same platforms. We just use them in a slightly more regulated fashion where we need to ensure reconstructability of absolutely everything that we do. So in a clinical trial scenario, if you imagine the subject in the top middle of the page, they are being dosed with an antibody or a drug or a novel chemical entity. And ultimately what happens is we get blood from those patients, and then we analyze the concentration of the drug in the blood, and then anything that has happened in terms of a pharmacodynamic endpoint associated with that drug pathway. And that could either be a cytokine or chemokine. We can measure that by Luminex or the MSD platform. Could be an immunological endpoint where it switches on specific markers on the surface of an immune cell. We can measure that by flow cytometry. And we can then culture cells and identify whether the drug has actually altered the way that cells behave in a very sophisticated manner. So basically what we do is to pick out that drug and what the drug is doing in the clinical trial scenario to provide the sponsor with enough data to fulfill their package to the regulators of how the drug PK profile is measured and how the drug has actually behaved within that patient population. So, what is the current sort of workflow? Well, generally to engage with Agilex, we generally evaluate the clinical trial synopsis or the protocol that's generated by the sponsor and the CRO, which gives an idea of exactly which tests are required, the sampling time points, and we then generally have a teleconference with the sponsor during the RFP phase to get a little bit more information, and then we put a proposal together, which ultimately, once signed, we then generate the methods. So we generally tend to develop methods from scratch or utilizing some of the IP generated in the sponsor's lab. And we develop all of these methods to meet the EMA, FDA, and RCH regulatory bioanalytical guidances, which means that the data that we generate is accepted globally at all of the regulatory agencies, and we validate these assays to meet these regulatory guidances. And so this schematic on the screen just provides a typical snapshot of how long it takes to actually get these assays developed. And the critical part really is for us to have these assays developed ahead of when that drug first goes into the first patient or first volunteer. So normally we're looking at about 12 weeks back from that first patient, first dose is when we need to be in the lab setting these assays up to ensure that we have enough stability data, and we've also looked at the collection. Some of the analytes can be pretty sticky, and we want to ensure that any collecting vessel, any tube that the blood is being collected in, or urine or CSF, that drug doesn't bind to the plastic where that the matrix is being collected, to ensure that we are measuring the drug appropriately for that clinical setting. So I hope that I've been able to provide, in this very short time frame, a little bit of a snapshot of, what we do. My contact details are here on this end slide. Please feel free to email and contact us for any further information, and we'd be happy to provide some additional information for you. And I thank you very much for your attention, and happy to answer any questions.
Thank you, Kurt. Thank you for the interesting presentation. I've been engaged with a couple of your colleagues, and every time I have a conversation with Agilex, I see new opportunities how we can further integrate with our LA Biomed City Innovation District. I do have a question here, and that's, what are the main considerations for sponsors for assay setup?
So one of the main considerations is really, by the time we get to the clinical phase of the cycle, the sponsor would have done a lot of work preclinically within a GLP model. And one of the considerations is the level of sensitivity that we need for those assays for the clinical phase of the study. Generally, for a preclinical study, you dose very high levels in that animal because you're looking at safety, and you're looking at pushing the limits of safety of those new molecules. When you go into the human population in the clinic, you start out at very low levels, which often means that we need a very sensitive assay. So that is essentially the first question we normally ask is, what level of sensitivity do you need for that first dose cohort? And then in terms of critical reagents, it's specific for the biological drugs coming to market, is having specific antibodies against that drug that we can use to create a ligand binding assay. So we're reliant really on things that are commercially available or reagents the sponsor lab has produced themselves. And we need these to be as pure and specific as possible in order for us to be able to develop an assay that is incredibly highly specific to pick out only the sponsor drug in that serum or plasma of that patient that has a whole variety of inflammatory mediators that oftentimes impact the assay and cause a lot of noise. So those are the general considerations, for that, that we're asking right at the beginning, and then also considerations around the study design, which generally needs to mirror what the sponsor had done in that animal population in order for us to be able to use, our learnings from the preclinical to move us forward to the clinical situation.
And following up on that then, so how far in advance should those assays be set up for a clinical trial?
So it really depends on whether it's a small or a large molecule in terms of regulated bioanalysis. Small molecule assays can be set up in a lot faster time frame. Biologics in general are a bit more complex because you're dealing with antigen-antibody interactions and specificity and selectivity issues in matrix, and it does take a bit longer. But generally, you're looking at about 12 weeks before first patient, first dose, to have a method set up and then validated to the EMA and FDA bioanalytical guidances, and all of that data through quality assurance and some stability data in order to have the assay ready for deploying onto those very precious patient samples that is part of the clinical trial.
Thank you, Kurt. I really like to thank you for your interesting presentation again. And it's now time to move on to the second speaker. It's a pleasure to welcome Mark Roe, CEO and founding partner of Fusetec. Mark is an entrepreneur by nature. Everybody who knows him recognizes, and he really enjoys solving global problems. Mark's last adventure, Fusetec, was commenced to address the global problem of how we train surgeons. So I would like to welcome Mark and look forward to hearing more about the innovations and the opportunity Mark brings to Adelaide, because they are significant, I believe.
Thank you very much, Yvette, for a kind introduction. I too would like to pay my respects to the traditional custodians, past, present and future. Fusetec was founded in response to the research question, how do surgeons train? Historically, surgical training has been a bit problematic on cadavers and living patients. I often find myself pondering at what point in training is a resident confident and competent to perform a complex surgery. So Fusetec's mission is to improve healthcare outcomes by providing safe, affordable and effective training alternatives to upskill residents and continually advance surgical training. Now, if we go back to 2018, Adelaide University, the FES courses there were run with cadavers. November 2019, technology had advanced that they started using Fusetec products rather than cadavers. So I'll just quickly show this 30-second video. So here we are in Adelaide University Wet Lab. We have 80 surgeons, 40 endoscopic stacks. We have, I believe on this occasion, we supplied 160 training models for the same price as 20 cadavers the year previously. So the surgeons effectively got 8 times the amount of surgical training, and each model had a different anatomy in it. So the teacher, PJ Wormer, would teach model number 1, then everybody in the class would do model number 1. That's basically how our technology rolls. So moving forward, why we do what we do, there's a lot of unmet global needs in surgical training. Firstly, 40% of residents lack their confidence in their ability even after 5 years of training. There's 143 million additional surgical procedures are needed annually at low to middle income countries. Greater than 1 million shortage of surgeons and anaesthetists in low to middle income countries. So Fusetec is designing products that can easily ramp up or that can assist in training surgeons. I'll just show this video, another 30-second video. This is a snippet of what we've been doing during COVID, where we haven't been able to have face-to-face meetings. This again is in collaboration with Adelaide University, Professor Aoki Basaltis and Professor Wormel, doing surgical training in Japan, based from Adelaide University. As you can see, surgical training now can be done remotely. Us shipping models all the way around the world. We show them how to do the dissection here, and then it's done on the other side of the world under surgical guidance. The next quick video I'm about to share you is progression from our traditional surgical anatomical models to now we're developing tumours and pathologies. So this model here is a transnatal, transphenoidal approach to a pituitary tumour. And it doesn't sound like there's any sound, so he's actually, this Professor Alkis is removing a concha bullosa there, creating, and he's about an hour into the surgery now. Looks like he's about to incise the mucosa and delineate it from the septum, creating a mucosal flap. The flap is now being tucked away, and I can see the sphenoid opening in the background. The sphenoid ostium, I should say. So the sphenoid ostium has been opened first superiorly towards the skull base, and here we are in now looking at the, that would be the sphenoid bone, and with a carotid artery on both sides of that. So he's going to remove the mucosa from the sphenoid bone. He's now drilling away the sphenoid bone, being very thin, so taking a lot of precaution here not to damage the dura mater. we can now see the bulk has been drilled away, and I think that's a back bite. He's removing the last of it, and you can see the soft dura mater behind it in the skull base. He's now putting an incision in the dura mater. Looks like he's going parallel to the carotid. He should now open that and make a, I think it's called a dura flap. Okay, and this is the tumour sitting behind this, the dura mater here. So he's sucking it out initially, and got a few little stubborn bits there, so you'll have to cut them off somehow. Okay, scraping them out. Oh, he's hit the carotid artery there, so this is a problem, but it's good. It's only a training model. Sucking the blood away, and he'll now apply a muscle patch. And in real life, you have only about 4 minutes, I believe, till morbidity sets in. So the professor's working quickly here. I can see the bleeding slowing now. Now he's getting the mucosal flap and covering this sphenoid opening as he's exiting the surgery. The next video goes about Professor Alke talking about the experience, which I'll just go past. So Fusetec products, as you can see on the right-hand side, a whole bunch of ticks. So they're fully operable with all surgical instruments, anatomically accurate, multiple pathologies, pathology on demand, etc. No ethical issues. We can FedEx them around the world. So the advantage of it is particularly in a lot of countries like the MENA region, where it's very difficult to get cadavers. This is one way to train surgeons with pathology on demand. You know, Fusetec is embracing technology where the potential to advance surgical training can be learnt in a classroom without risk of harmful bacteria, the need for wet lab, or risk to stakeholders, all due to technological advances. As you have seen, even training can now be done remotely. So we're doing our best to advance surgical training and provide tools for the surgeons and residents. We're certainly open to collaborations with universities, hospitals and surgeons, or organisations who share common interests. We have multiple research projects on the go with materials, software, anatomy, and we're also doing multiple publications at the moment. Thank you very much for allowing me to speak a little bit about Fusetec, and hope we can collaborate with you guys soon.
Thank you, Mark. I think great you've included those videos, made it very tangible. It also reminded me why I didn't choose to be a medical professional. So please feel free to ask questions. I have a question for you. I think Fusetec is bringing quite interesting niche capabilities and partners into our innovation district. And I'm just curious, are there any other areas you're keen to explore further development in? Or is it really focused on the surgical, or do you see other opportunities you're at least exploring based on the capabilities that are already available within Fusetec?
We're certainly open to all opportunities. Where our main focus is on surgical training. My partner, John, is focusing at the moment on prosthetics. We're also in communication with numerous companies in space and defence, but I allocate that to various people. I don't want to distract from my focus. What we're also doing is not only developing the products, but we're planning to launch later this year our moving into Adelaide Biomed City, straight across the road from Samri into 21 North Terrace. So that's a very exciting move for us as our company is expanding and growing into the region. So we're very much looking forward to that.
And we're looking forward to celebrating that milestone, Mark. Thank you both speakers, industry speakers, for the fantastic presentations today. I don't know if you're aware, but you're the first industry presenters within this mini review series. Until now, we all had researchers present. And the reason we're really starting to integrate more industry speakers here is because research industry collaboration is really key to drive further innovation and commercialization. So thank you to be the first one today. If you're interested, if you're listening and you're interested to speak in those mini reviews, feel free to reach out to us. You can contact us through the website. If you have any feedback now, we will keep this webinar open for a couple of more minutes. So feel free to add your feedback in the Q&A. We always appreciate that. And next Tuesday, there's another mini reviews. It focuses on patient-reported outcome measures, PROMs. Mr. Jared O'Brien will talk about the statewide patient-reported measure program. And Associate Professor Rosanna Tavella will present patient-reported outcome measured in the CADOSA registry. So thank you for joining us today, and we hope to welcome you again next week.